Tonight's lecture is over chapter 26 concerning the urinary system. And this chapter begins on page 1042 in your textbook. The urinary system contributes to homeostasis by excreting wastes, alternating blood composition, pH, volume, and pressure, maintaining blood osmolarity, and producing hormones. The urinary system consists of 2 kidneys, 2 ureters, 1 urinary bladder, and 1 urethra. And nephrology is the scientific study of the anatomy, physiology, and pathology of the kidneys. The branch of medicine that deals with the male and female urinary systems and the male reproductive system is called urology. A physician who specializes in this branch of medicine is called a urologist. Moving on to the next slide, this is figure 26.1 on page 1043. This shows the organs of the urinary system in the female. So urine is formed by the kidneys, then it passes first into the ureters, then into the urinary bladder for storage, and finally through the urethra for elimination from the body. The kidneys regulate blood volume and composition. They help regulate blood pressure, pH, and glucose levels, and they also produce hormones, calcitrol and erythropoietin. And they also excrete wastes in the urine. The ureters, on the other hand, transport the urine from the kidneys to the urinary bladder. And then the urinary bladder will store the urine until it's expelled into the urethra. And the urethra discharges the urine from the body. So some of the homeostatic kidney functions include the regulation of blood ionic composition. And most importantly, this includes sodium ions, potassium ions, calcium ions, and phosphate ions. The kidneys accomplish this task by adjusting the amounts of these ions that are excreted into the urine. Kidneys also help with the regulation of blood pH. In this regard, the kidneys excrete a variable amount of hydrogen ions into the urine and conserve bicarbonate ions, which are an important buffer of hydrogen ions in the blood. Both of these activities help to regulate blood pH. The kidneys also help to regulate blood volume. The kidneys adjust blood volume by conserving or eliminating water in the urine. An increase in blood volume increases blood pressure. A decrease in blood volume decreases blood pressure. Speaking of blood pressure, the kidneys also help to regulate blood pressure. They do this by secreting the enzyme renin, which activates the renin-angiotensin-aldosterone pathway. Increased renin causes an increase in blood pressure. Moving on to the next slide, the kidneys also help to maintain the osmolarity of the blood. They do this by separately regulating the loss of water and the loss of solutes in the urine. The kidneys maintain a relatively constant blood osmolarity close to 300 milliosmoles per liter. Kidneys are also involved in the production of hormones. The kidneys produce 2 hormones. That's calcitrol, the active form of vitamin D, which helps regulate calcium homeostasis. And you can refer back to chapter 18 for that. And they also produce erythropoietin, which stimulates the production of red blood cells. Kidneys also help with the regulation of the blood glucose level. So similar to the liver, the kidneys can use the amino acid glutamine in gluconeogenesis, the synthesis of new glucose molecules. They can then release glucose into the blood to help maintain a normal blood glucose level. So moving on to the next slide, this is figure 26.2 on page 1045. And this shows the position and coverings of the kidneys. So the kidneys are what we call retroperitoneal, and they are partly protected by the lower ribs. Furthermore, The kidneys are surrounded by a fibrous capsule, a pararenal fat capsule, and the renal fascia. And this next slide is the same figure, but now we're looking at a paramedian plane of the kidney. So the kidneys are a paired, reddish, kidney bean-shaped organ located just above the waist between the peritoneum and the posterior wall of the abdomen. Because their position is posterior to the peritoneum in the abdominal cavity, the organs are said to be retroperitoneal. The kidneys are located between the levels of the last thoracic and third lumbar vertebra, a position where they are partially protected by ribs 11 and 12. If these lower ribs are fractured, they can puncture the kidneys and cause a significant, even life-threatening type of damage. The right kidney is slightly lower than the left kidney because the liver occupies considerable space on the right side superior to the kidney. So this cartoon can be seen at figure 26.3 on page 1046. And this shows the indented area that is called the renal hilum. A typical adult kidney is 10 to 12 centimeters long and 5 to 7 centimeters wide, and approximately 3 centimeters thick. That's about the size of a bar of bath soap and has a mass of about 135 to 150 grams. The concave medial border of each kidney faces the vertebral column. Near the center of the concave border is an indentation called the hilum of the kidney, through which the ureter emerges from the kidney along with blood vessels, lymphatic vessels, and nerves. Next slide. Concerning the external layers of the kidney, 3 layers of tissue surround each kidney. The deep layer, the fibrous capsule, is a smooth, transparent sheet of collagen-rich connective tissue that is continuous with the outer coat of the ureter. It serves as a barrier against trauma and helps maintain the shape of the kidney. The middle layer, the pararenal fat capsule, is a mass of fatty tissue surrounding the fibrous capsule. It also protects the kidney from trauma and holds it firmly in place within the abdominal cavity. And finally, the superficial layer, the renal fascia, is a collagenous and elastic, dense, irregular connective tissue that anchors the kidney to the surrounding structures and to the abdominal wall. On the anterior surface of the kidneys, the renal fascia is deep to the peritoneum. Moving on to the internal renal anatomy. So if you take a coronal section through the kidney, this will reveal 2 distinct regions: a superficial light red region called the renal cortex, and a deep, darker reddish-brown inner region called the renal medulla. The renal medulla consists of several cone-shaped renal pyramids. The base, or the wider end of each pyramid, faces the renal cortex, and its apex, the narrow end, is called a renal papilla, and this points toward the renal hilum of the kidney. The renal cortex is a smooth, textured area extending from the fibrous capsule to the bases of the renal pyramids and into the spaces between them. It is divided into an outer cortical zone and an inner juxtamedullary zone. Those portions of the renal cortex that extend between the renal pyramids are called renal columns. And so this slide brings us back to figure 26.3 on page 1046, and we're just looking a little more closely at the inner anatomy of the kidney. So we see the 2 main regions of the kidneys. The one is superficial, the light red region called the renal cortex, and then the deep dark red region called the renal medulla. And if we look on the right, we can see a cadaveric view of that. So moving on to the blood and nerve supply of the kidneys. Because the kidneys remove waste from the blood and regulate its volume and ionic composition, it is not surprising that they are abundantly supplied with blood vessels. Although the kidneys constitute less than 0.5% of the total body mass, they receive 20 to 25% of the resting cardiac output via the right and left renal arteries. Many renal nerves originate in the renal ganglion and pass through the renal plexus into the kidneys along with the renal arteries. The renal nerves are part of the sympathetic part of the autonomic nervous system, and they help regulate blood flow through the kidneys. Moving on to the nephron, this figure is on page 1049, figure 26.5, and it shows that nephrons are the functional unit of the kidneys. And in this particular cartoon, we're looking at the structure of nephrons and the associated blood vessel. Note that the collecting duct and the papillary duct are not part of the nephron. Furthermore, the nephron consists of two parts, a renal corpuscle and a renal tubule. Moving on to the next slide. So in the renal corpuscle, blood plasma is filtered into the renal tubule. Moving on to the next slide, the two components of a renal corpuscle are the glomerulus and the glomerular capsule, or the Bowman's capsule. The Bowman's capsule is a double-walled epithelial cup that surrounds the glomerular capillaries. Blood plasma is filtered in the glomerular capsule, and then the filtered fluid passes into the renal tubule, which has three main sections. Those three sections are the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. And if we look at the next slide, we find this cartoon on page 1052, figure 26.6. And this cartoon shows a great detail of the renal corpuscle, including its glomerulus and its glomerular capsule. And on the right, we view a light micrograph of the renal corpuscle. Moving on, the glomerular endothelial cells have large pores or fenestrations and are very leaky. The basal lamina lies between the endothelium and the podocytes. The podocytes form pedicles between which are filtration slits. And if we look at the next slide, we can see a filtration membrane. This cartoon appears on page 1055, and this is figure 26.8. So during glomerular filtration, water and solutes pass from blood plasma into the capsular space. So at number 1, the glomerular endothelial cells are leaky because they have large fenestrations. The size permits all solutes in blood plasma to exit the glomerular capillaries, but prevents the filtration of blood cells. They're located among the glomerular capillaries and in the cleft between the afferent and efferent glomerular arterioles. Secondly, the basement membrane is a porous layer of acellular material between the endothelium and the podocytes that consists of minute collagen fibers and negatively charged glycoproteins. The pores within the basement membrane allow water and most solutes to pass through. However, the negative charges of the glycoproteins repel blood plasma proteins because they are anionic. The repulsion hinders the filtration of these proteins. Thirdly, we see the slit membrane between the pedicels. Extending from each podocyte are thousands of foot-like processes termed pedicels that wrap around the glomerular capillaries. The space between the pedicels are the filtration slits. A thin membrane, the slit membrane, extends across each filtration slit and permits the passage of molecules having a diameter smaller than 0.006 to 0.007 micrometers, and this includes water, glucose, vitamins, amino acids, and very small blood plasma proteins, ammonia, urea, and ions. Less than 1% of the albumin, the most plentiful plasma protein, passes through the slit membrane because with a diameter of 0.007 micrometers, it is slightly too big to get through. Moving on to the next slide, about 80 to 85% of the nephrons are the cortical nephrons. Their renal corpuscles lie in the outer portion of the renal cortex, and they have a short nephron loop that lies mainly in the renal cortex, and it penetrates only into the outer regions of the renal medulla. And they create urine with the osmolarity similar to blood. So this cartoon is also part of figure 26.5, and this is on page 1050, and this shows the cortical nephron and its vascular supply. So if you look at the tan box there, you can see the flow of fluid through a cortical nephron. It starts at the glomerular capsule. It moves into the proximal convoluted tube, then into the descending limb of the nephron loop, and then into the ascending limb of the nephron loop, and then into the distal convoluted tubule. And this will drain into the collecting duct. So the other 15 to 20% of nephrons are called juxtamedullary nephrons. Their renal corpuscles lie deep in the renal cortex, close to the renal medulla, and they have a long nephron loop that extends into the deepest regions of the renal medulla. Long nephron loops receive their blood supply from the peritubular capillaries and from the vasa recta that arise from the efferent glomerular arteriole. In addition, the ascending limb of the nephron loop of the juxtamedullary nephrons consists of two portions, a thin ascending limb followed by a thick ascending limb. These nephrons enable the kidney to secrete very concentrated urine. This cartoon is on page 1051, and it's still part of figure 26.5, and this shows a cartoon of the juxtamedullary nephron and its vascular supply. And if we look at the tan box, we can see the fluid flow through a juxtamedullary nephron, where similarly to the cortical nephron, it begins with the glomerular capsule, moves into the proximal convoluted tubule, then moves into the descending limb of the nephron loop, then into the ascending limb of the nephron loop, and then into the thick ascending limb of the nephron loop, and then into the distal convoluted tubule, which will drain into a collecting duct. Moving on to the next slide concerning the juxtaglomerular apparatus, the ascending loop contacts the afferent arteriole at the macula densa. The wall of the arteriole contains smooth muscle cells called the juxtaglomerular cells. The apparatus regulates blood pressure in the kidney in conjunction with the autonomic nervous system. So this cartoon is on page 1054. This is figure 26.7, and this shows the relationship of a nephron's structure to its three basic functions, which are glomerular filtration, tubular reabsorption, and tubular secretion. Excreted substances remain in the urine and subsequently leave the body. Glomerular filtration occurs in the renal corpuscle. Tubular reabsorption and tubular secretion occur along the renal tubule and the collecting duct. So step 1 is the first step of urine production. Water and most solutes in blood plasma move across the wall of the glomerular capillaries, where they are filtered and move into the glomerular capsule and then into the renal tubule. At step 2, which is called tubular reabsorption, filtered fluid flows through the renal tubules and through the collecting ducts. The renal tubule cells reabsorb about 99% of the filtered water and many useful solutes. The water and solutes return to the blood as it flows through the peritubular capillaries and the vasa recta. Note the term reabsorption refers to the return of substances to the bloodstream. The term absorption, by contrast, means the entry of new substances into the bloodstream or into the body as it occurs in the digestive canal. And then at step 3, we call this tubular secretion. So the filtered fluid flows through the renal tubules and collecting ducts. The renal tubule and the duct cells secrete other materials such as wastes, drugs, and excess ions into the fluid. Notice that tubular secretion removes substances from the blood. Moving on to the next slide, glomerular filtration. The fluid that enters the capsular space is called glomerular filtrate. The fraction of blood plasma in the afferent glomerular arterioles of the kidneys that becomes glomerular filtrate is the filtration fraction. This glomerular filtration is driven by blood pressure, opposed by capsular hydrostatic pressure and blood colloid osmotic pressure. Also, water and small molecules move out of the glomerulus. And in one day, about 150 to 180 liters of water pass into the glomerular capsule. Moving on to the next slide, the glomerular filtration rate is the amount of filtrate formed by both kidneys each minute. Homeostasis requires kidneys to maintain a relatively high glomerular filtration rate. If the glomerular filtration rate is too high, substances pass too quickly and are not reabsorbed. And if the glomerular filtration rate is low, nearly all reabsorbed and some waste products are not adequately excreted. And this next slide is on page 1057. This is figure 26.9. And this shows that the pressures that drive glomerular filtration. Taken together, these pressures determine the net filtration pressure. Glomerular blood hydrostatic pressure promotes filtration, whereas capsular hydrostatic pressure and blood colloid osmotic pressure oppose filtration. So to that end, the glomerular filtration depends on 3 main pressures. 1, the glomerular blood hydrostatic pressure is the blood pressure in the glomerular capillaries. 2, the capsular hydrostatic pressure is the hydrostatic pressure exerted against the filtration membrane by fluid already in the capsular space in the renal tube. And thirdly, the blood colloid osmotic pressure is due to the presence of proteins such as albumin, globulins, and fibrinogen in the blood plasma, and it also opposes filtration. So moving on to the next slide, we see that concerning the glomerular filtration rates, the averages are different between males and females, where we see the males are filtering more milliliters per minute than the females are. And this glomerular filtration is controlled by renal autoregulation, neural regulation, and hormonal regulation. So moving on to the next slide concerning renal autoregulation, the kidneys themselves help maintain a constant renal blood flow and glomerular filtration rate despite normal everyday changes in blood pressure like those that can occur during exercise. This capability is called renal autoregulation and consists of 2 mechanisms. One is the myogenic mechanism, and the second one is tubuloglomerular feedback. The myogenic mechanism occurs when stretching triggers the contraction of smooth muscle fibers in the walls of afferent glomerular arterioles. As the blood pressure rises, the glomerular filtration rate also rises because the renal blood flow has increased. Furthermore, when arterial blood pressure drops, the smooth muscle fibers are stretched less and thus relax. The afferent glomerular arterioles dilate, renal blood flow increases, and the glomerular filtration rate increases. The myogenic mechanism normalizes renal blood flow and the glomerular filtration rate within seconds after a change in blood pressure. The second contributor to renal autoregulation is tubuloglomerular feedback. This is named because part of the renal tubules, the macula densa, provides feedback to the glomerulus. When the glomerular filtration rate is above normal due to elevated systemic blood pressure, fluid flows more rapidly along the renal tubules. As a result, the proximal convoluted tubule and nephron loop have less time to reabsorb sodium ions, chloride ions, and water. The macula densa cells are thought to detect the increased delivery of sodium ions, chloride ions, and water, and to inhibit the release of nitric oxide from cells in the juxtamorular apparatus. Because nitrous oxide causes vasodilation, afferent glomerular arterioles constrict when the level of nitric oxide declines. As a result, less blood flows into the glomerular capillaries, and the glomerular filtration rate decreases. When blood pressure falls, causing glomerular filtration rate to be lower than normal, the opposite sequence of events occurs, although to a lesser degree. Tubuloglomerular feedback operates more slowly than the myogenic mechanism. So here's figure 26.10 on page 1058, and this shows a negative feedback loop of the tubuloglomerular feedback mechanism. See, the macula densa of the juxtamoglomerular apparatus provide the negative feedback regulation of the glomerular filtration rate. So moving on to the next slide concerning the neuroregulation of the glomerular filtration rate. Like most blood vessels of the body, those of the kidney are supplied by the sympathetic autonomic nervous system fibers that release norepinephrine. Norepinephrine causes vasoconstriction through the activation of alpha-1 receptors, which are particularly plentiful in smooth muscle fibers of the afferent glomerular arterioles. At rest, sympathetic stimulation is moderately low, the afferent and efferent glomerular arterioles are dilated, and the renal autoregulation of the glomerular filtration rate prevails. With moderate sympathetic stimulation, both afferent and efferent glomerular arterioles constrict to the same degree. Blood flow in and out of the glomerulus is restricted to the same extent, which decreases the glomerular filtration rate only slightly. With greater sympathetic stimulation, however, as occurs during exercise or hemorrhage, vasoconstriction of the afferent glomerular arterioles predominates. As a result, blood flow into the glomerular capillaries is greatly decreased and the glomerular filtration rate drops. This lowering of renal blood flow has two consequences. It reduces urine output, which helps conserve blood volume, and it promotes greater blood flow to other body tissues. And moving on to hormonal regulation of the glomerular filtration rate. So two hormones contribute to the regulation of the glomerular filtration rate. Angiotensin II reduces the glomerular filtration rate, and atrionatriuretic peptide increases the glomerular filtration rate. Angiotensin II is a very potent vasoconstrictor that narrows both afferent and efferent glomerular arterioles and reduces renal blood flow, thereby decreasing the glomerular filtration rate. Cells in the atria of the heart secrete atrionatriuretic peptide. So the stretching of the atria occurs when blood volume increases, and this stimulates the atrionatriuretic peptide hormone by causing the relaxation of the glomerular mesangial cells. Atrionatriuretic peptide, or ANP, increases the capillary surface area available for filtration. So the glomerular filtration rate rises as the surface area increases. And here we're looking at table 26.2 on page 1059, and this summarizes the regulation of the glomerular filtration rate. So I will let you review this table on your own time. This next slide is part of the same table. This slide is also part of the same table. So moving on to the tubular reabsorption and secretion. This is on page 1059. So much of the filtrate is reabsorbed, especially water, glucose, amino acids, and ions. And furthermore, secretion helps to manage pH and rid the body of toxic and foreign substances. So this is table 26.3 on page 1060, and it summarizes the substances filtered, reabsorbed, and secreted per day. So I'll let you go ahead and go over this table on your own time. And on this next slide, it's just a continuation of table 26.3, so please review this. Okay, so moving on to reabsorption routes. Fluid can leak between the cells in a passive process known as paracellular reabsorption. Even though the epithelial cells are connected by tight junctions, the tight junctions between cells in the proximal convoluted tubes are quote-unquote leaky and permit some reabsorbed substances to pass between cells in the peritubular capillaries. In some parts of the renal tubule, the paracellular route is thought to account for up to 50% of the reabsorption of certain ions and the water that accompanies them via osmosis. And then we have the transcellular reabsorption, and this is where a substance passes from a fluid into the tubular lumen through the apical membrane of a tubule cell across the cytosol and out into the interstitial fluid through the basolateral membrane. So this cartoon is on page 1060, and this is figure 26.11. And this shows the reabsorption routes, the paracellular reabsorption and the transcellular reabsorption. So in paracellular reabsorption, water and solutes in the tubular fluid return to the bloodstream by moving between the tubule cells. And then in transcellular reabsorption, solutes and water in the tubular fluid return to the bloodstream by passing through a tubule cell. So moving on to transport mechanisms, when renal cells transport solutes out of or into tubular fluid, they move specific substances in one direction only. Not surprisingly, different types of transport proteins are present in the apical and basolateral membranes. The tight junctions form a barrier that prevents the mixing of proteins in the apical and basolateral membrane compartments. The reabsorption of sodium ions by the renal tubules is especially important because of the large amount of sodium ions that pass through the glomerular filters. So if we look at this slide, we see that we'll start with the primary active transport. So in primary active transport, the energy derived from the hydrolysis of ATP is used to pump a substance across a membrane. The sodium-potassium pump is one such pump. Then, in secondary active transport, the energy stored in an ion's electrochemical gradient, rather than the hydrolysis of ATP, drives another substance across the membrane. Secondary active transport couples the movement of an ion down its electrochemical gradient to the uphill movement of a second substance against its electrochemical gradient. So symporters move the substances in the same direction, and antiporters move the substances in the opposite direction. Moving on to the next slide, solute reabsorption drives water reabsorption because all water reabsorption occurs via osmosis. About 80% of the reabsorption of water filtered by the kidneys occurs along with the reabsorption of solutes such as sodium ions, chloride ions, and glucose. Water Reabsorbed with solutes in the tubular fluid is termed obligatory water reabsorption because the water is obliged to follow the solutes when they are reabsorbed. This type of water reabsorption occurs in the proximal tubule and the descending limb of the nephron loop because these segments of the nephron are always permeable to water. Reabsorption of the final 20% of water is termed facultative reabsorption. The word facultative means capable of adapting to a need. Facultative water reabsorption is regulated by antidiuretic hormone and occurs in the late distal tube and throughout the collecting duct. Moving on to the next slide, reabsorption and secretion in the proximal convoluted tubule. Most solute reabsorption in the proximal convoluted tubule involves sodium ions. Sodium transport occurs via symport and antiport mechanisms in the proximal convoluted tubule. Normally, filtered glucose, amino acids, lactic acid, water-soluble vitamins, and other nutrients are lost in the urine. Rather, they are completely reabsorbed in the first half of the proximal convoluted tubule by several types of sodium ion symporters located in the apical membrane. In another secondary active transport process, the sodium ion, hydrogen ion antiporters carry filtered sodium ions down their concentration gradient into a proximal convoluted tubule as hydrogen ions are moved from the cytosol into the lumen. This causes sodium ions to be reabsorbed into the blood and hydrogen ions to be excreted into the tubular fluid. The proximal convoluted tubule cells produce hydrogen ions needed to keep antiporters running in the following way. Carbon dioxide diffuses from the peritubular blood or the tubular fluid or is produced by metabolic reactions within the cells. Furthermore, solute reabsorption in the proximal convoluted tubules promotes osmosis of water. Each reabsorbed solute increases the osmolarity, first inside the tubule cell, then in the interstitial fluid, and finally in the blood. Water thus moves rapidly from the tubular fluid via both the paracellular and transcellular routes into the peritubular capillaries and restores osmotic balance. In other words, reabsorption of solutes creates an osmotic gradient that promotes the reabsorption of water via osmosis. Cells lining the proximal convoluted tubule and the descending limb of the nephron loop are especially permeable to water because they have many molecules of aquaporin 1. This integral protein in the plasma membrane is a water channel that greatly increases the rate of water movement across the apical and basolateral membranes. So this cartoon shows both figures 26.12 and 26.13 on page 1062 of your textbook. On the left-hand side, we see the reabsorption of glucose by sodium ion glucose symporters in the cells of the proximal convoluted tubule. Normally, all filtered glucose is reabsorbed in the proximal convoluted tubule. And then on the right-hand side of the slide, we see the actions of the sodium ion, hydrogen ion antiporters in the proximal convoluted tubules. So there at cartoon A, we see the reabsorption of sodium ions and the secretion of hydrogen ions via the secondary active transport through the apical membrane. And then on the second cartoon, letter B, we see the reabsorption of bicarbonate ions via the facilitated diffusion through the basolateral membrane. The sodium ion, hydrogen ion antiporters promote the transcellular reabsorption of sodium ions and the secretion of hydrogen ions. And on this cartoon, we're looking at page 1063. This is figure 26.14, and it shows the passive reabsorption of chloride ions, potassium ions, calcium ions, magnesium ions, urea, and water in the second half of the proximal convoluted tube. So electrochemical gradients promote the passive reabsorption of solutes via both paracellular and transcellular routes. Moving on to the next slide concerning the reabsorption in the loop of Henle, or we could call this the loop of the nephron, or the nephron loop, because all of the proximal convoluted tubules reabsorb about 65% of the filtered water. Fluid enters the next part of the nephron loop, at a rate of about 40 to 45 milliliters per minute. The chemical composition of the tubular fluid is now quite different from that of the glomerular filtrate because glucose, amino acids, and other nutrients are no longer present. Plus, the osmolarity of the tubular fluid is also still close to the osmolarity of blood. The nephron loop reabsorbs about 15% of the filtered water, 25% of the filtered sodium ions, potassium ions, and calcium ions, 35% of the filtered chloride ions, 10% of the filtered bicarbonate ions, and a variable amount of filtered magnesium ions. Furthermore, the apical membranes of cells in the thick ascending limb of the nephron loop have sodium ion, potassium ion, and chloride ion symporters that simultaneously reclaim one sodium ion, one potassium ion, and two chloride ions from the fluid in the tubular lumen. And we can see this cartoon on page 1064, figure 26.15. This shows the sodium ion, potassium ion, and two chloride ion symporter in the thick ascending limb of the nephron loop. So cells in the thick ascending limb of the nephron loop have symporters that simultaneously reabsorb one sodium ion, one potassium ion, and two chloride ions. So moving on to the next slide concerning the reabsorption in the early distal convoluted tubule. So in this case, fluid enters a distal convoluted tubules at a rate of about 25 milliliters per minute. This is because about 80% of the filtered water has now been reabsorbed. The early or initial part of the distal convoluted tubule reabsorbs about 5% of the filtered sodium ions and 5% of the filtered chloride ions. Reabsorption of the sodium ions and chloride ions occurs by means of the sodium ion chloride ion symporters in the apical membranes. Plus, sodium-potassium pumps and chloride ion leakage channels in the basolateral membranes then permit reabsorption of the sodium ions and chloride ions into the peritubular capillaries. The early distal convoluted tubule is also a major site where the parathyroid hormone stimulates reabsorption of calcium ions. The amount of calcium ion reabsorption in the early distal convoluted tubule varies depending on the body's needs. Moving on to the next slide concerning the late distal convoluted tubule and the collecting duct. By the time the fluid reaches the end of the distal convoluted tubule, about 90 to 95% of the filtered solutes and water have returned to the bloodstream. So the principal cells and the intercalated cells are present at the late or terminal part of the distal convoluted tubule and throughout the collecting duct. The principal cells reabsorb sodium ions and secrete potassium ions. These cells also have receptors for aldosterone and antidiuretic hormone. The intercalated cells reabsorb sodium bicarbonate and secrete hydrogen ions. Thereby, they play a role in blood pH regulation. In addition, the intercalated cells reabsorb potassium ions. In the late distal convoluted tubules and collecting ducts, the amount of water and solute reabsorption and the amount of solute secretion varies depending on the body's needs. So this is figure 26.17 on page 1066, and this shows the negative feedback regulation of facultative water reabsorption by antidiuretic hormone. So most of the water reabsorption, or 90% of it, is obligatory and 10% of it is facultative. So this table, if it is in the book, I can't find it. But this is the summary of the distal convoluted tubule on the collecting duct. And it looks like it also provides information on the proximal convoluted tubule. So I'm leaving it in the PowerPoint presentation. Please review this table on your own time. And this table is also a part of the previous slides, so please study this on your own time. I think it has some good information for you. And then this table is on page 1067, table 26.4. And this is a summary of hormonal regulation of tubular reabsorption and tubular secretion. So again, with all these tables, I leave this up to you to review it on your own time. And this slide is part of the same table. And this slide is also part of the same table. Remember, this is on page 1067 in your textbook. So urine production begins on page 1067. So even though your fluid intake can be highly variable, the total volume of fluid in your body normally remains stable. The homeostasis of body fluid volume depends in large part upon the ability of the kidneys to regulate the water loss in urine. Normally function kidneys produce a large volume of dilute urine when the fluid intake is high and a small volume of concentrated urine when the fluid intake is low or there is large amount of fluid loss. Furthermore, antidiuretic hormone controls whether dilute urine or concentrated urine is formed. In the absence of antidiuretic hormone, urine is very dilute. However, a high level of antidiuretic hormone stimulates the reabsorption of more water into the blood, producing concentrated urine. So moving on to the next slide. A high intake of urine results in the dilute urine of high volume, and a low intake of fluid results in the concentrated urine of low volume. Seems like that makes sense from a physical point of view. Moving on to the next slide. Concerning the formation of dilute urine, the glomerular filtrate has about the same ratio of water and solute particles as blood. So that means it has a similar osmolarity. However, tubular osmolarity changes due to a concentration gradient in the medulla. So moving on to the next slide, when dilute urine is formed, the osmolarity in the tubule does three things. First of all, it increases in the descending limb, then it decreases in the ascending limb, and it also decreases more in the collecting duct. Moving on to the next slide, cells lining the thick ascending limb of the loop have symporters that actively reabsorb sodium ions, potassium ions, and chloride ions from the tubular fluid. These ions pass from the tubular fluid into the thick ascending limb cells, then into the interstitial fluid, and finally some diffuse into the blood inside the vasa recta. And although some solutes are being reabsorbed in the thick ascending limb, the water permeability of this portion of the nephron is always quite low, so water cannot follow by osmosis. As solutes, but not water molecules, are leaving the tubular fluid, the osmolarity drops. The fluid entering the distal convoluted tubule is thus more dilute than plasma. Furthermore, while fluid continues flowing along the distal convoluted tubule, additional solutes, but only a few water molecules, are reabsorbed. The early distal convoluted tubule cells are not very permeable to water and are not regulated by antidiuretic hormone. Finally, the principal cells of the late distal convoluted tubules and collecting ducts are impermeable to water when the antidiuretic hormone level is very low. Thus, tubular fluid becomes progressively more dilute as it flows onward. By the time the tubular fluid drains into the renal pelvis, its concentration can be very low. This is 4 times more dilute than blood plasma or glomerular filtrate. Moving on to the next slide, this is figure 26.18 on page 1068, and this outlines or gives us a cartoon of the formation of dilute urine. So the numbers in this cartoon indicate osmolarity in millimoles per liter. The heavy brown lines in the ascending limb of the nephron loop and the distal convoluted tubule indicate impermeability to water, and the heavy blue lines indicate the last part of the distal convoluted tubule and the collecting duct, which are impermeable to water in the absence of antidiuretic hormone. And then the light blue lines, the light blue areas around the nephron represent interstitial fluid. So when antidiuretic hormone is low, urine is dilute and has an osmolarity that's less than the osmolarity of blood. So concerning the formation of concentrated urine, when water intake is low or water loss is high, the kidneys must conserve water while still eliminating waste and excess ions. Under the influence of antidiuretic hormone, the kidneys produce a small volume of highly concentrated urine. Urine can be 4 times more concentrated than blood plasma or glomerular filtrate. So countercurrent multiplication is the process by which a progressively increasing osmotic gradient is formed in the interstitial fluid of the renal medulla as a result of concurrent flow. Concurrent multiplication involves the long nephron loops of the juxtamedullary nephrons. So in this case, solutes are pumped out of the ascending limb, but water stays in the tubule, and therefore the medulla osmolarity is increased. Moving on, in the presence of antidiuretic hormone, the collecting ducts become very permeable to water. The tubular fluid there becomes very concentrated, and then the movement of water also carries urea into the medulla, contributing to its osmolarity. Moving on to the next slide, we already talked about countercurrent exchange. This is where the loop and duct cells require nutrients and oxygen from the blood supply. The capillaries that feed them, the vasa recta, form loops like those of the nephron loops in the medulla. The incoming and outgoing blood will have a similar osmolarity, and this maintains the medulla concentration gradient. And now we're looking at the cartoon on figure 26.19, page 1069. This shows the mechanism of urine concentration in long loop juxtamedullary nephrons. The green line indicates the presence of sodium ions, potassium ions, and 2 chloride ion symporters that simultaneously reabsorb these ions into the interstitial fluid of the renal medulla. This portion of the nephron is also relatively impermeable to water and urea. All concentrations are in milliosmoles per meter. So again, this cartoon is outlining the formation of concentrated urine, and the formation of concentrated urine depends on a high concentration of solutes in the interstitial fluid in the renal medulla. So at step 1, the symporters in the thick ascending limb cells of the nephron loop cause a buildup of sodium ions and chloride ions in the renal medulla. At step 2, we see the countercurrent flow through the descending and ascending limbs of the nephron loop, and that establishes an osmotic gradient in the renal medulla. At step 3, the cells in the collecting ducts will reabsorb more water and urea. And then at the 4th step, urea recycling causes a buildup of urea in the renal medulla. and this creates more concentrated urine. So moving on to the next slide, the evaluation of kidney function begins on page 1072. Routine urinalysis primarily evaluates for the presence of abnormalities in the urine. So I'd like for you to turn to page 1073 and view table 26.6. And table 26.6 goes through all of these bullet points here. So the albumin, glucose, red blood cells, ketone bodies, and microbes are all outlined in detail in that table. So please review that on your own time. Moving on to the next slide. Urine transportation and storage begins on page 1075. From collecting ducts, urine drains into the minor calyxes, which joins to become the major calyxes that unite to form the renal pelvis. From the renal pelvis, urine first drains into the ureters and then into the urinary bladder for storage. Urine is then discharged from the body through the single urethra. So each ureter transports urine from the renal pelvis by the peristaltic waves, hydrostolic pressure, and gravity. There's no anatomical valve at the opening of the ureter to the bladder. Then when the bladder fills, it compresses the opening and prevents backflow. The bladder is a hollow, distensible muscular organ with the capacity averaging about 700 to 800 milliliters. And men will have larger bladders than women because the female reproductive organs require them to have smaller bladders. And this is figure 26.21 on page 1075. This shows the ureters, the urinary bladder, and the urethra in a female. So as I said, urine is stored in the urinary bladder before being expelled by micturition, and micturition is just a fancy word for urination. Moving on to the next slide, micturition. The discharge of urine from the urinary bladder, called micturition, is also known as urination or voiding. Micturition occurs via a combination of involuntary and voluntary muscle contractions. When the volume of urine in the urinary bladder exceeds 200 to 400 milliliters, pressure within the bladder increases considerably, and stretch receptors in its wall will transmit nerve impulses to the spinal cord. These impulses propagate to the micturition center in the sacrospinal cord segments S2 and S3 and trigger a spinal reflex called the micturition reflex. In this reflex arc, parasympathetic impulses from the micturition center propagate to the urinary bladder wall and internal urethral sphincter. The nerve impulses cause contraction of the detrusor muscle and relaxation of the internal urethral sphincter muscle. Simultaneously, the micturition center inhibits somatic motor neurons that innervate skeletal muscle in the external urethral sphincter. On contraction of the urinary bladder wall and relaxation of the sphincters, urination takes place. It should also be noted that in males, the urethra also discharges semen as well as urine, and to that end, it would follow, or we could deduce that males will have larger or longer, I should say, ureters than females do. So this cartoon is on page 1076, figure 26.22, and this makes a comparison between the male and female urethras. So the male urethra is about 20 centimeters in length, while the female urethra is about 4 centimeters in length. So I kind of said that on the previous slide, that the males have a longer urethra than the females do. So moving on to waste management in other body systems, and this begins on page 1079. Body buffers. Buffers in body fluids bind excess hydrogen ions, thereby preventing an increase in the acidity of body fluids. However, they do have limits. Blood. The bloodstream provides pickup and delivery services for the transport of wastes. The liver. The liver is the primary site for metabolic recycling. The lungs. With each exhalation, the lungs excrete carbon dioxide and expel heat and a little water vapor. Sweat glands. Especially during exercise, sweat glands in the skin help eliminate excess heat, water, and carbon dioxide, plus small quantities of salts and urea. In the digestive canal, or the GI tract, through defecation, the digestive canal excretes solid, undigested foods, wastes, some carbon dioxide, water, salts, and heat. The development of the urinary system begins on page 1080. And during the third week of fetal development, a portion of the mesoderm begins to differentiate into the kidneys. And in this cartoon on page 1080, figure 26.23, we can see the development of the urinary system. Moving on to aging in the urinary system. This is on page 1080. With aging, the kidneys shrink in size, have decreased blood flow, and filter less blood. These age-related changes in kidney size and function seem to be linked to a progressive reduction in blood supply to the kidneys as an individual gets older. Moving on to the next slide. This focus on homeostasis is found on page 1082 of your textbook. So the urinary system contributes to all body systems. For example, the kidneys help regulate volume, composition, and pH of body fluids by removing waste and excess substances from the blood and excreting them in urine. And the ureters transport urine from the kidneys to the urinary bladders, which stores it until it is eliminated through the urethra. And I'll let you go through all of the other points on this slide on your own time. Moving on to the next slide, homeostatic imbalances. Renal calculi, or kidney stones. The crystals of salt present in urine occasionally precipitate and solidify into insoluble stones called renal calculi or kidney stones. Urinary tract infections. This term is used to describe either an infection of part of the urinary system or the presence of a large number of microbes in the urine. Glomerular disease. A variety of conditions may damage the kidney glomeruli, either directly or indirectly because of disease elsewhere in the body. Renal failure. This is a decrease or cessation of glomerular filtration. Polycystic kidney disease is one of the most common inherited disorders. PKD in the kidney tubules becomes riddled with hundreds or thousands of cysts. In addition, inappropriate apoptosis of cells in non-cystic tubules leads to a progressive impairment of renal function and eventually to the end-stage renal failure. Urinary bladder cancer. Each year, nearly 12,000 Americans die from urinary bladder cancer. It generally strikes people over 50 years of age and is 3 times more likely to develop in males than females. Kidney transplant. A kidney transplant is the transfer of a kidney from a donor to a recipient whose kidneys no longer function. In the procedure, the donor kidney is placed in the pelvis of the recipient through an abdominal incision. And cytoscopy. This cytoscopy is a very important procedure for the direct examination of the mucosa of the urethra in the urinary bladder and prostate in males. And that concludes our lecture for this evening.